In the first section of Chapter 25, we're going to focus on DNA structure and replication. The structure and replication of DNA has a lot of history behind it. In 1952, Alfred Hershey and Martha Chase did an experiment, and they demonstrated through this experiment that DNA is the genetic material, not proteins. In the two experiments, the results were that viral DNA labeled with P32 was found in bacteria, not in the medium, so it showed that it had entered the bacteria, and the viral protein in the capsids labeled with the S35 found in the medium were found in the medium and not in the bacterium, so it didn't enter the bacteria. Only the DNA entered bacteria. Here we have a diagram that's going to help explain this a little bit better. So first of all, they put P32, labeled the DNA plasmid within the virus, and this is the capsid of the virus made out of protein, and this is the DNA. And when the virus attaches itself to the bacteria, it injects its DNA into the bacteria. They put them in the blender, put the bacteria in the blender, and it knocked the capsids off. Then they centrifuged the solution, and that allowed for the bacteria to go to the bottom and the viruses to come to the top because of size and mass, and they found that the bacteria was radioactive, so therefore the DNA had gone into the bacteria. On the other hand, they took S32 and labeled the capsid with it. So this time, not the DNA was radioactive, but the protein capsule around it was. Again, they put them into a blender and they agitated them and knocked the viruses loose from the bacteria. And when they put them in a centrifuge, then separated the bacteria from the virus capsids, they found that the radioactivity was in the virus capsids, so at the top this time instead of at the bottom. And you can see the difference here in this. So what this shows is that it's not protein that goes into the bacteria with the DNA, it's just the DNA. And that's what they showed through this experiment. So in 1953, Watson and Crick received a Nobel Prize for coming up with the structure of the DNA molecule. DNA is a chain of nucleotides, and each nucleotide is made up of 3 subunits: the phosphate group, the sugar group, or the deoxyribose in this case, and a nitrogen-containing base. Those 4 bases are adenine, guanine, thymine, and cytosine. We refer to them as A, G, T, and C. If we're talking about the types of chemical group they belong in, adenine and guanine are purines, thymine and cytosine are pyrimidines. The way you can keep that straight is the thymine and cytosine both have Y's, pyrimidines both have Y's in them, so you can tell them apart. Now when they complementary base pair, they base pair not identical but complementary, and a purine is always going to go with a pyrimidine. Adenine always pairs with thymine in DNA. Guanine always pairs with cytosine. This double helix is the common picture that you see for DNA. So you see the structure and you automatically think DNA. And if I were to unravel that, to unwind it, it looks like the stairs of a ladder. And we have adenine across from thymine, guanine across from cytosine every time. I want you to notice that there's a phosphate group, a sugar, and then the nucleotide base, thymine in this case. We have the sugar, the phosphate group, and adenine. And these are held together by these dashed lines represent hydrogen bonds. So 2 hydrogen bonds between thymine and adenine, 3 between guanine and cytosine. We also have a 5' end and we have a 3' end from this direction. And if you'll notice, those primes come from the location of the carbons around the sugar. So we have the 5' end is what's hooked to the phosphate group on both sides. So the 5' end is down with the phosphate. That means the 3' end is up. 5' end is up, 3' end is down. We're going to talk about the replication of DNA. When we talked about mitosis, we talked about this happening during the S phase of interphase. So we want to look and see what really happens during replication of DNA. The process is called semi-conservative. And the reason it's semi-conservative is it's not like putting a paper into a copy machine and having an old copy and then your new copy come out, or an original and a new copy. Each daughter DNA molecule is made up of 1 new chain of nucleotides and 1 from the parent DNA molecule. The 2 daughter DNA molecules are going to be identical to the parent. So you have to know the steps of replication. Before replication begins, the two strands of the parent molecule are hydrogen bonded together. The enzyme DNA helicase unwinds and unzips the double-stranded DNA. The new complementary DNA nucleotides fit into place along the separated strands by complementary base pairing. These are positioned and joined by DNA polymerase, which is another enzyme. DNA ligase seals any breaks in the sugar phosphate backbone. And the two double helix molecules are then identical to each other and to the original DNA molecule. So we see this happen in this strand. This is a replication fork, and DNA helicase has come in to the parent molecule and has unzipped that hydrogen bonding through there, and we have new nucleotide pairing to get an old strand and a new strand, and an old strand of the daughter DNA double helix. Parental DNA molecule contains so-called old strands, and that hydrogen bonding is what holds those together. The region of replication, the parent DNA is unwound and zipped. The new nucleotides are paired with the old ones. Replication is complete. A double helix is composed of an old strand and a new strand, or a parental strand and a daughter strand. Today's lecture was just over the replication of DNA, and I do expect you to know for the test the steps that happen in DNA replication. Remember the link on YouTube to the song that helps us remember what those stages are, and study it, memorize it. It's just one of those things that you're going to have to put into applications. We'll do a lab that has to do with it so you can visualize it. But it is one of the essay questions on the test is explain DNA replication, and I expect you to be able to do that.